Okay, everyone, welcome to today's episode on this monthly seminar, African seminar. The title of today's seminar will be Image Analysis using Open Software. And of course, we all know image analysis has been a fundamental part of biological research, which we cannot even do without again. So in this seminar, we'll focus on the use of open software for image analysis, especially Fiji. Some people call it image, but it's still the same. So the speaker for this monthly seminar will be Dr. Giselle Miranda. In 2019, she received a double PhD degree in computer science and applied mathematics from the University of Sao Paulo, Brazil, and in applied biological sciences from Ghent University in Belgium. This is an excellent combination of interests, which makes her a perfect fit for today's seminar. She is a Chan Zuckerberg imaging scientist and a researcher at KTH Royal Institute of Technology in Sweden. And so she's also attached to the life cell imaging facility at the Carolina Institute. And Carolina Institute is one of the top research centers in Europe. And quite interestingly, she is a Chan Zuckerberg Award winner. I mean, it's one of the competitive initiative grant in Europe in life sciences. She's also an active member of bioimage informatics facilities at KTH and the life cell imaging facility at the Carolina Institute. And she's been having a keen interest in the intersection of computer science and biology. And she works on multidisciplinary research projects in the biomedical field involving computer vision, machine learning, and data analysis. Without further ado, I would like to invite Dr. Giselle Miranda to the podium, to the meeting. Now it's over to you, please. You can share your screen and comments.
Okay, so hello everyone. Thanks to Toom for the introduction. It's my pleasure to be here with you today talking about image analysis and software tools for image analysis. So I'm Giselle. I'm a bioimage analyst in the bioimage informatics facility and at the lifestyle imaging facility at Karolinska. And today I prepared a presentation where I'm going to start by just describing a bit what we do at the facility, what are our typical projects. Then we are going to a more fundamental part with some basic concepts and how to ensure image quality for image analysis. And then we are going to spend most part of this presentation talking about image analysis workflows, and followed by some examples in Fiji, and finally some concluding remarks. And you can interrupt me at any time for questions. So, well, basically, you just presented part of my CV there. So I'm from Brazil. I got my bachelor in at the University of Sao Paulo in biomedical informatics. So since then, I started working at the intersection between computer science and biology. I did the master in the same university, and it's when I started working with microscopy images. I also had experience in the private sector as a software developer, this time working with radiology images. And then I did my PhD between Brazil and Belgium in computer science and applied math, and in biological and applied sciences. And then in 2019, I joined KTH, the Royal Institute of Technology, as a researcher and working mainly as a bioimage analyst in the facility. So here is we can see University of Sao Paulo. This is the state of Sao Paulo in Brazil. This is where I was working with Professor Bruno. And in Belgium, I also spent some time in Belgium. Actually, I'm in Ghent right now for a short period. And then, so when I went to Sweden, I joined the team of the bioimage informatics facility, which is one of the core facility of research infrastructure called the Science for Life Laboratory. And we are connected to the bioimaging and the bioinformatics platform. So we help users in Sweden to perform image analysis pipeline. So we try to bridge the gap between their user needs and the available tools and technologies. Here is our website for projects. If you're interested to know more, I'm going to show some examples soon. This is how I was used to work before COVID. Now after COVID, I work more online. And we are connected with also international groups like the Eurobioimaging, the Chan Zuckerberg Initiative, and the NeoBias, which is the network of the bioimage, network of the European bioimage analysts. And it's quite interesting. I'm going to leave also some links at the end. They are very much engaged in education in image analysis. So they have also material and a great YouTube channel about software tools for image analysis. So starting with the concepts now. So what is bioimage analysis? How we can define it? So I'm used to say that it's the process of understanding and quantifying microscopy and medical image data. So usually we have sets or groups of images from different experimental conditions, and we want to extract information from them. We want to compare them. So we want to quantitative analyze those images. So probably 20, 25 years ago, we could do a visual analysis or even a manual analysis, but nowadays it's practically impossible. So we need tools that help us to quantify the information or extract features in the images. And this is due mainly to the advanced microscopy techniques, generating huge amounts of data, like light sheet microscopy, electron microscopy. We also have more sensitive fluorescent labels and sensors leading to multi-channel images. Higher throughput data creation. So we are talking about higher nanometer resolution, volumes, 3D images, and time lapses. But at the same time, now we have increased the storage capacity and faster computer and better algorithms to deal with such data. So we are here at the intersection between computer science, which provides us with tools, computational tools to handle such data, statistics, to analyze such data, and it's also necessary a good knowledge of biology to understand the underlying process, biological processes. So let's give a look in some examples. And some of these examples are projects that I've been working in the context of the facility. So here we have, for instance, nuclei detection in H&E image. This is a breast cancer tissue. And here the aim was to integrate spatial transcriptomics data, gene expression data with image analysis data. When it comes to tissues, we also work this is a project where we had to analyze the structure of the epithelial tissue. So this is a multi-channel immunofluorescence image of cervical tissue used in research on sexually transmitted infections. And the goal here was to analyze the layers of the epithelial tissue. We can also talk about time series. So in this project, this is a time series of beating cardiomyocytes. And the goal here was to study the reactions of these cells to different pharmacological agents. And how did we do that? So here what we can see is the calcium waves. So first we segmented the cells and then we analyzed the calcium waves using frequency analysis. Here's another example of time-lapse image data, where the goal here was to study the neurite outgrowth, the dynamics of neurite outgrowth. So here we have different steps of this pipeline, including, for instance, nuclei detection and cell identification. And finally, we have an example here of 3D segmentation in electron microscopy. So segmentation is the process of separating the objects of interest in the image. And when it comes to 3D, it's even more challenging. So these are just a few examples what bioimage analysis is, and of course, it goes beyond. And I hope I have motivated you to start this talk. So now we go to a more fundamental part. So we have a common nomenclature here when we talk about image analysis workflows. So we are dealing with digital images, and it's important to know or remember that an image can be simply defined by a matrix of numbers. So if we have an image, if we zoom in a specific region, we will see the pixels or the pictures elements, which assume specific values. So our image is in fact a 2D matrix with a certain matrix with a certain number of rows and columns, and assuming specific values. Where do the pixel values come from? They come from the photosensitive devices attached to the microscopes responsible for converting the light signal into digital signal. And the range is what we call the bit depth, which leads to the number of gray values that we can represent. So this is an important property of a digital image. And we calculate the number of gray values as 2 to the power n, where n is the bit depth. So for instance, if we have 2 bits, then we can represent only black and white. But as we increase the number of bits, then we can represent more information. If we consider 8-bit images, then we can represent 256 values. Microscopy images are usually 12 or 16 bits. So the key concept here is as more bits you have, more details you can represent, more information you can represent. It's also important to keep in mind the concept of histogram, which is the distribution of gray levels. So through the histogram, we can retrieve, for instance, basic statistics of an image. Here we can see that this is an 8-bit image, since the range of gray values is going from 0 to 255. Also, another important concept is brightness and contrast. Brightness is a relative term. So if we analyze images A and B, we can see that B is brighter than A, and it also has a higher contrast. And we can see these through the histogram. So in A, the range of values go from 0 to 166. And in B, it occupies the whole range of the possible values, gray levels. So we say that B has a higher contrast, and it's also brighter. Also important to keep in mind the color information. So we can have RGB images coming from stained tissue. This is a section of a tissue stained with hematoxylin and usine. And here now our matrix is a 3D matrix where we have information of the 3 channels, red, green, and blue. But in the case of fluorescent images, the color is just a color coding. So for instance, here we have a 3-channel image. And for each channel, we have a specific matrix. And the color information here is just for visualization purposes. So this leads us to our next concept, which is the lookup table, which is a color coding of your image. So for instance, if we take the green channel of our previous example, we can represent, we can change the visualization in such a way that it's easier to visualize some structures. So maybe you can highlight the brightest regions of your image, like here, for instance. And important to note that this doesn't change the original value. So when you change the lookup table, the original values are not modified. And why this is where it can be used? It can be used, for instance, for visualizing the segmentation results, or for identifying saturation. So this is a special lookup table called the high-low, where you can see the areas that are saturated, both in the max value in red and in the minimum value in blue. Also, another property of images, it's good to keep in mind the dimensions of the data that you are working with. So you can have, for instance, 2D images where you have X and Y dimensions, or we can have stacks in the case of 3D images. So here we have X, Y, and Z dimensions. Or you can even have what we call a hyperstack. So these are images that have, besides the 3 dimensions, also 2 channels and a time lapse. So this is a 5D image example of a mitosis. So we have the time lapse as well. So once we have our image, how to guarantee, how to ensure image quality for image analysis. So some image artifacts, they cannot be corrected by image analysis. So for instance, saturation is one example, as we are going to see here. So here's a comparison of the same image with different histograms. So here we have an underexposed histogram, which leads to a very dark image, and here an overexposed histogram of very bright regions. And this is also not good because we lose information about the contour of the objects of the image, even some details about the bolt. So it's good, the ideal situation is when you have a well-exposed histogram, avoiding saturation both in the minimum values and maximum values. Bleed-through is also another artifact that can compromise our analysis. So here in the first line, we have an example of bleed-through between the green and the red channels. So here, if you aim at colocalization analysis, it's compromised because they are already colocalized, but it's a false colocalization. So this would be the ideal situation. So here we don't have. So it should also be avoided. There's not much we can do from the analysis part. About file formats, it's good to keep in mind that we have the microscopy file formats. So for instance, we have ND2 for Nicole or CZI for Zeiss, and usually they are readable with specific software, but we have libraries, for instance, in Fiji that allows us to read these formats. And we also have general file formats. JPEG should not be used for scientific image because due to compression, TIFF is a good format, and we can also represent 3D images with TIFF. And PNG should only be used to transfer and display, so no stacks is possible here. So choose the right format, check the export options, and never save an image, an original image in lossy compression formats. Also, it's good to keep in mind about bit conversion when you're dealing with images. So in general, increasing the bit depth does not change the pixel value. So when you go from 8-bit to 16-bit, but when you go the other way around, scale can or not be applied to pixel values. So you are changing, if you're scaling, you are changing the pixel values. And also some operations can be reversible. So when changing brightness and contrast, for instance, if saved, so in Fiji, for instance, that's the Fiji interface, you can apply the transformation. So always a good practice is to always create a copy of the original image when working in Fiji. However, some things can be corrected with image analysis. It would be better if they're corrected when acquiring the image. So let's see some examples. So denoising is one of them. So here we have an example of a noisy image, and here the corrected, the filtered image. What we can also do as a pre-processing is also a registration or a stabilization of time series to correct, in this case, to correct a drift in the time series. So this is also possible to do. And illumination correction. So, well, every optical system is subject to distortions. And I think that maybe the most common effect here is the vignetting effect, which leads to uneven illumination. Here we can see the mosaic So we can see the borders or the tiles. And this here we have the corrected version, and here as well. So here's an example of illumination correction. So these are examples of what can be done with image analysis to improve the quality of your images. So it's good to see, it's good to keep in mind that image analysis, your image analysis goals has to be included in your experimental setup, going from sample preparation until the analysis. So how you can optimize our sample preparation to also get the optimal analysis. Okay, so now we can talk a bit about image analysis workflows. So what we have here is what I would like to talk about is what is a typical workflow in image analysis. These steps may vary, but in general we have a pre-processing step, which involves like background subtraction, noise reduction, followed by segmentation, which is the separation of the objects of interest in the image, and a connected component analysis of the post-processing, which is a refinement of the segmentation, and finally image quantification. So let's see each of these steps in detail. By pre-processing, we can apply filters, for instance, in order to, they can be designed to modify or enhance image properties such as edges, corners, blobs. So here we have an example of a noisy image containing two nuclei. And so this is the unfiltered image, and here it's our filtered image, and this is obtained by what we call a convolution between the original image and our kernel, which is designed to, for instance, in this case, we have the average filter. So for a specific neighborhood defined by the kernel size, we will average the pixels will be averaged, and the new value will be defined for the filtered image. But this is done for each pixel in the original image. So if we now if we go to the next pixel, the 43 in the unfiltered image, we again compute the average, and then which is approximately 12 also. So the effect of this filter is to smooth the image. And here we can compare, for instance, similarly, we have the median filter that instead of the average takes the median value of this neighborhood. So first we have to sort it and then take the median element. It's like in the statistics when we compute the median for a specific set of values. And the median filter is especially useful when we have this situation, what we call a salt and pepper noise, because we have a region that is predominantly dark and a single pixel here that is white. So we want to filter this out. And if we apply the mean filter, what will happen is that these values will be averaged. But if we apply the median, this value will be filtered out, because it's probably the last, it is the last element of the, is the highest value of this neighborhood. So the choice of the filter depends on what you want to do, what is your goal. So let's give a look now at the result of the median filter applied to our HeLa cells example for different kernel sizes. So here's a radius of 2, here 4, and here 6. So we can see how also the size of the neighborhood affects the final result. And again, this is the size of your neighborhood should be chosen based on the size of the particles that you want to filter. So it's also a parameter to be tuned in your pipeline. Filters can be used also not only for denoising, but also for edge detection or to blur the image. So there are different types of filters. These are known as convolution filters, but we also have filters, for instance, that work in the frequency domain and so on. So here, these are examples of convolution filters, but there are different types of filters. So once we've filtered our image, and usually here now, in this case, we apply the median filter. Now we have a more homogeneous region inside each nuclei, and this helps segmentation. So segmentation is the process of separating the objects of interest from the background. Thresholding methods are the simplest approach to segmentation. And thresholding means defining a pixel value above which we have our particles, our objects of interest. And below this value, we have the background. So here in white, we have the segmented objects, and in black, we have the background. And you can also create also a labeled mask using a lookup table to better visualize the results. Here is an example in Fiji. You can define this value, the threshold value, manually, but we have different methods described in literature on how to implement the threshold. Fiji has about 16 of these methods. And we can see that different methods lead to different results. So for instance, this is more restrictive than these. So here we have less, the regions that are segmented are smaller than here. Selecting the threshold manually is not, in general, not reproducible. So what is recommended is to choose one threshold algorithm and stick to it the whole study. So which method to use is also something that has to be attuned as the parameter of the filter. Okay, so once we have our binary mask, we can see that we can still improve this mask. So we have some holes here that can be eliminated. So this is the post-processing, so or the what can be improved after the segmentation. So if we apply a few holes operation, now we can obtain the whole nuclei of these three particles. We still do not have this. This nucleus is not complete here. We are still losing some part here, but for these three, we could improve. One thing that we can do here is filter this particle out by circularity. So the way here we have, for instance, these three that are more rounded shape than this one. So if you don't want to extract measurements of these nucleus here, we can filter it out by circularity. Since it's not complete, it's not. It might not be meaningful. The measurements extracted here. So the post-processing means tuning a bit, improving the binary mask. Another thing that can be done as post-processing is often we have this situation where we have touching objects, and in fact we want separated objects here. And here's an example of the watershed transform. There are different types of watershed transform. For gray level images, binary images. Here we are using the Euclidean, the one based on Euclidean distance map. And the idea is that to generate this distance map where we have from the boundary of the nucleus, in this case of the nucleus, in the direction of the center, the distance increases. So it's like filling two catchment basins, and when they meet each other, then a watershed line is created. Here is the same, is a similar animation using an example from the Fiji website. And now we have two objects. So we can summarize our workflow then by filtering, thresholding, post-processing the mask, and finally quantifying our objects. So here we have measures related, here we have area, mean, standard deviation, mean, and max. But we have, we can have different groups of measures. So we can have measures related to intensity, measures related to the shape, so shape descriptors as circularity, for instance, and so on. So we have different types of measures that can be extracted. This is an example also of the available measures in Fiji. So this is what we call a typical workflow using a thresholding segmentation plus filtering. We also have approaches based on machine learning for segmentation, more recent, but it's being more used now, gaining more attention and solving problems that are difficult to be solved with more classical workflows, let's say with thresholding. So here's what we call a supervised paradigm. So we have an input data set of annotated training data. So the particles that we want to analyze, they are already annotated. They are marked in this image. And then a neural network is trained, and we don't have to specify filter in this case. So we just need to input annotated training data. And the network will build a model for the segment, in this case, for the segmentation of the particles. So we will get results, what we call instance-based segmentation. So each particle here is identified. The good thing is that, and here usually you need a lot of training, of annotated training data. But if you can generalize in such a way that your model, when you present a new image to this trained model, then you will get your output, your segmentation output. There are two methods that are considered state-of-the-art for cell nuclei segmentation. They are based on the learning approach, which is CellPulse and StarDist. CellPulse is a more generalist algorithm because it was trained on a data set with different types of images, of microscopy images, tissues, stained tissue, fluorescent images, and so on. And what is trained by the network is this flow representation that is generated for each particle. So here we have an example of, this is the input image, and this is the final segmentation. StarDist is a similar approach, but it was trained on H&E, stained tissue, and fluorescent nuclei. So here is a specific for nuclei, or you can even, if you have cells, rounded shape objects, it's you can also test the trained model. And we are going to see an example in Fiji. There is a plugin for StarDist in Fiji. Unfortunately, there's no plugin in Fiji for CellPulse, but you can run CellPulse in a Python script, or even there is a graphical interface to test your image with CellPulse. And both of them work for 2D and 3D. Another interesting tool is also here based on machine learning also. This is a plugin for Fiji called Trainable Weka Segmentation. And the idea here is that you specify, you make some annotations in your image. Here's an electron microscopy image. And we have basically three classes, the red, green, and blue. And you specify some, you mark some parts of this image representing each of these three classes. And what you get at the end is something like this, where you have the three classes segmented. So this approach is also quite interesting for image segmentation. I can briefly here mention some software tools. Of course, there are more, they're not limited to these lists, but I would say that Fiji ImageJ is one of the most important tools that we have for bioimage analysis, image analysis as well, more general. Cell Profiler, Elastic. So Elastic is similar to the trainable Weka segmentation that we just saw. We have QPath for tissue analysis, Napari for, it's a tool in Python. It's a good visualization tool, and you can also implement plugins and more custom solutions, libraries like ITK, OpenCV, and so on. So now I would like to show how to run this workflow in Fiji. So I will shift here to Fiji now. And I have here, so I don't know if you're familiar with Fiji. So we have here the main interface. You can ask questions if you want. And here we have a set of menus and some basic tools here for annotation. Here I have a data set of 5 images. So we have of nuclei. So we can see, we can have an idea of the variability of these images. And we can, for instance, so first I would like to filter, right, to get a more uniform distribution of the pixel intensities inside each nucleus. And we can do that using the process menu. So here I will apply the median filter that we saw in the presentation. Here we can have, before applying, we can also have a preview of the effect. So here if I increase the radius, then the effect will be higher. So I think I will start with 2. Oh, and a good approach that when working in Fiji, always keep a copy of the original image. You can duplicate an image from the image menu, but I will open again the original image. So now we have original image, filtered image. And this will be our mask. So now I will apply a threshold. And we can see here that depending on the method that we choose, we have different segmentations. So this looks like a good one. We don't have any holes in the masks. And then I will choose this one and apply. So now we have a binary image. We can see here in the Fiji interface the pixel values when we the mouse is over the image. So the background 0 and the white, the particles 255. So this is an 8-bit image. And now what we can do here, we can see that probably this, oh, sorry, I opened the 4th one. Let me open the 2nd one. Yeah, now we have the same image, sorry. Yes. So now it would be interesting that to separate these nuclei. So we can do this using the watershed transformation. And now we can see that the nuclei were splitted. So in this case, probably this shouldn't be split. But these 3 worked well, and here as well in the bottom. So we will have a smaller particle here. So what we can do now is when we analyze the particles, so Fiji has a tool called Analyze Particles to label each of these segmented particles. So what we can do is filter the smaller particles. So then we can eliminate these particles here. So for instance, here is in pixel squared. So I want to eliminate all the particles with area below 3000. And I also want to exclude the cells on the edge because we only have part, nuclei, we only have part of the nuclei there. So we cannot rely on the measurements extracted from these nuclei. So then if we run the Analyze Particles tool, now we can see that each selected particle is now labeled, right? And we can also show the overlays over the original image to compare or to check the quality of our segmentation. And also now we can first define the measures that we want. So here we can define in the Analyze menu what are the measures that we want, for instance, shape descriptors, area, mean wear value, the centroid pixels of each particle, the centroid pixel. And once we define, then we can measure. And here we have the results table. So for each particle, we have the selected measures, which can then be saved for future analysis, for instance. Okay, so this was done for one image, right? But if we have 100 images, what do we do? In Fiji, we have, we can create a script to automatize this analysis, like this one. So here I have an example of a pipeline. So this was written in the Fiji macro language, and this helps us to automatize the process for our whole data set. So here I have a script that basically does what we just did using the graphical interface of Fiji. Apart from some details here, you can see that the mean size of the nuclei is a parameter. Here is the command that runs the median filter. Here is the command for the threshold, and so on. So when I run this script, I just have to select my folder of interest, and then the analysis will be performed for each image. And here we have the log with the files that were processed. So here we can check the output. So in this pipeline, I'm also saving the output. So for each image, we have the segmentation results, the labels that were also exported, and the CSV file corresponding to the measurements for each image. And the last thing I would like to show is, I mentioned about Stardist. So Fiji has this, has some plugins that come with Fiji when you download, for instance, or you can also install additional plugins. And here we have the Stardist plugin. I can show an example of the segmentation for one of these images that we have here. So here we just have to first open the image and then run the Stardist plugin. So as I mentioned, it was trained on fluorescent nuclei and H&E images. So here's a fluorescent nuclei, and you can run it. It has some two important parameters, in fact, that control the overlap. In this case, for clustered cells or nuclei, and the probability threshold. I will run with the optimized parameters from the training, and we can compare the results with using the typical workflow. So again, here, when you run, you have the ROI manager, and you can choose to show the segmentation results. And again, this is a plugin, sorry, that is macro recordable, what we call. So you can also include, here it's the command that runs the Stardist, and you can also include this plugin in your script, so you can run it for your whole data set. And here we can already observe the results. So here I have the output using the Stardist plugin. So maybe here, yeah, it was not a good result here. And again, even with the machine learning approaches, we can pre-process our image filtering before applying the method, and even post-processing. So here we can also filter particles by size. And now just to finalize a bit, some minutes late, but what are the learning outcomes of this presentation? I wanted to show some examples of what can be done with image analysis, the limitation of each example, also understand what is made by image segmentation, filtering, binary mask, and so on, and understand which image artifacts can be corrected by image analysis. So it's a field of its own, it's complex, but you can get help with that. Here are some useful links, some an active discussion forum. I don't know if you're familiar with forum.image.sc. A book on bioimage data analysis and on bioimage analysis workflows. Some interesting website with some trained models that you can run for your own data set, for instance, and a link to the New Bias Academy YouTube channel with also great videos and material about image analysis. And thank you. If you have any questions, please feel free.